Next, we have Dr. Duarte Toubarro, and he's going to present some research for us. So give him a hand.
Okay. First of all, thank you, Sandra, for this opportunity. Thank you, Mark, for your words. And this is a really pleasure to be here. It's amazing, amazing environment, and I learn a lot. It's really, really nice to be here. I don't have a donkeys, and I don't know nothing about donkeys. I'm a researcher in the University of Azores, and I'm invited by Marcos to do some research on milk to try to understand some molecular values underlying the milk of donkey. And today I will present something about the second challenge of Marcos. So the first challenge is to show something about the milk and the functional activity of milk. And the second challenge and where we face now is to produce kefir from donkey milk. That is challenging for Marcos. But the idea of the university is to understand what is there, what kind of bacteria we have there, and to talk about the profit, the probiotics that we can have in this kind of products. So first of all, the human body is close. In close relation with the complex ecosystem, and the microbiome means the collection of organisms, viruses, and bacteria, and that live in our body and on our body. Okay, and this microbiome start to be present in specific environments, really specific environments in our gut, in our mouth, in our skin, and became more and more specialized, and can play important roles, really important roles in physiology and assimilation of nutrients, protection against the pathogens, and the modulation of immune response. So they share a symbiotic relationship with us, and that part of microbiome is different from the microbiome that we eat this morning. So this is also the microbiome, but we have the balance between the good microbes and bad microbes. So how you get your microbiome? As soon as you burn, you are exposed to billions of microbes that take up the residence to our body, and they start to persist in our body during your life. And you take these microbes from your food, from your hills. You can change your microorganisms when you are sick or not. The medications that you are prescribed can change the microbiome, and even less the exercise, the animals that you contact, the persons that you contact can change your microbiome. And this microbiome doesn't stop it to change. So this is the really. But how to change the microbiome communities you can improve the health effect with these microbiome communities or not? Yes. So the very it's really difficult to say what is the normal community looks like, because it's a complex. It's a mix of different organisms, not just bacteria, not only bacteria, but also fungi and other microorganisms. So this is really, really difficult to say what is a community that is healthy. But one thing that we know is a balance between pathogens and the probiotic bacteria are really important. And this balance we must reach. Okay. So how to restore the balance of the microbiome, how to change the microbiome in the positive way. So you know you are what you eat, and you are also what you are not eating. Okay, so in case of the fermented dairies can help and is already known that can help a lot to improve the balance of the microbiome, because we have something that we called probiotic bacteria that have a positive effect on biotic. So kefir is a fermented milk. It's a fermented milk that is really a long tradition on Europe and east of Europe, and is a potential health association. So we have thousands of work on that subject. Here we have a resume. I don't know if it's possible to see a resume about 5,800 articles, scientific articles that review clinical studies with the use of therapeutic kefir as a therapeutic agent. Okay, the use of kefir as a therapeutic agent. So here we have a resume. Later on, I will share the presentation with you. Can check, and. Of course, we have articles related with the modulation of inflammation and, for example, improve the microbial environment of your gut after consumption of kefir, and this is what we are looking for. So that balance and usually are related with bioactive peptides and exopolysaccharides also that can have antimicrobial activity against some pathogenic bacteria that we have in the gut. However, the potential beneficial effects of the midgut microbiome is much more than that, and we know that we can look for the microbiome itself, not just for the peptides that are present in fermented milk or fermented donkey milk, but also the bacteria that are present on this live food. So in our lab, we start to look for the DNA, and we sequence the 3 samples of kefir from Marcos. And we try to analyze what we have there. We have about 3,100 sequence reads of DNA, and in that DNA we found about 57 OTUs. OTUs means unities, the taxonomic unities of bacteria in this case. Okay, so we have at least 57 different bacteria there. And what kind of bacteria we have? We have mainly lactobacillus. Lactobacillus are really important and have a long story of health related with lactobacillus. And depending of the process of fermentation, and Marcus gave me 3 different samples that are trying to optimize the process of fermentation. And depending of the process of fermentation, you have here different results. And as you saw, we have the same main bacteria. Lactobacillus are main in all the experiments that we did. Acetobacter is also really important, and are also important probiotics, and Lactococcus. There are 90% of bacteria that exist on kefir, in this case of kefir. This is a kefir is completely different from kefir that we obtain from cow milk. When we compare, I don't have this data here, but later on I can show you. When you compare the microbiome of kefir from cow milk with the microbiome that we obtain from donkey milk, are completely different. And are completely different because the raw material are completely different also, okay? As Zuzi showed this morning, we have lysozyme that is 6,000x more concentrated, that enzyme that kill bacteria, that is 6,000x more concentrated in donkey milk than in cow milk. So when I try to ferment something in that milk that have capability to kill bacteria, it's not easy. It's necessary an adaptation process, and some special bacteria, some special bacteria can resist. So this is the secret of that kind of kefir. And what we have lactobacillus, but we have a diversity of bacillus. I don't know if it's possible to see the diversity there. We have Lactobacillus kefiri, we have Lactobacillus kefiriensis, we have Lactobacillus lacticus, and also Acetobacter. But the Lactobacillus is the most important component of this probiotic product. So lactobacillus have a long story of recognition as safety, and is used as centuries as a food for food preservation, and as much attention as give to the antimicrobial compounds that this kind of bacteria produce. The bacteria is able to produce different antimicrobial compounds, but the acid lactic is the main compound that they produce and have a huge impact in microbiome. And this is really important because if we have a bacteria that is established in our gut and they are producing the lactic acid in situ, they start to modulate the communities that exist there. And this is really important to get the balance again, okay? And bacteriocins are other type of enzyme, of bacteria, of antimicrobial peptides that we have. But it's not all the story. That kind of bacteria, the lactobacillus, have the capability to touch, to bond to epithelial barrier and stay there adherent to the epithelium. Okay, so if they stay longer in the epithelium, and the reason why is they produce mucins and the exopolysaccharides that can touch very well to our epithelium. If they stay there, they start to produce metabolites that can also give us some positive effect. That butyrate, butyric acid. The butyric acid is something that is produced naturally by this kind of bacteria. And if they are attached in our gut, they start to produce in loco the butyric acid. That the butyric acid is, with the butyric acid, this is a fat acid, a small fat acid that can cleave and help to cleave the fibers and the fibers that we have in the food. So this is really important also for our nutrition. Okay, so this is a second layer of activity that we can find in bacillus, protecting attached to the epithelial barrier and protecting the epithelial barrier, but also the modulation of immune response. So this is a long story of articles that is already published with lactobacillus modulating the immune response. The question is, what kind of molecules the lactobacillus produce that modulate the immune system? And sometimes we don't know what kind of molecules exist, but we know, and we look for the pathways that are changed after the consumption of the lactobacillus, and many of them are related with immune system and immune modulation. The inflammation, JAK-STAT and GNK pathway, and are boosted by the presence of lactobacillus. Not all lactobacillus have the probiotic activity. Just some of them can have this activity. Okay? And this is the second part of the work. The first part is to know that they are there, who are there, and what kind of bacteria we have. But the second part is they are probiotic or not. To be probiotic, they should resist to bilic salts, should resist to really dramatic pH that we have in the stomach, like pH 1 or 2. And not all bacteria can resist to these conditions, and the ones that can be probiotic is the ones that resist to these conditions, okay? So we can screen the bacteria that can be a probiotic potential. Another screen that we can do is for the catalase. Catalase, this kind of bacteria produce mainly acid lactic, not catalase. So if we have isolates that are catalase negatives, means that are producing acid lactic and are something that we want. Okay. Other thing that we look for is diamylase negative. So we want a bacteria that can produce amount of enzymes, a number of enzymes, proteases, carbohydrate active enzymes, and so on. But we don't want a bacteria that can disrupt our cells. So this is another test that we did, and this is the screen of the hemolysis. Hemolysis, if the bacteria can disrupt our blood cells or not, and the positive effect is not, okay? And of course, the DNA is negative, so we have something that are attached to our epithelium and should be not produce DNases, so not degrade our DNA. Okay, so DNase negative is our test that we did, and gelatinases, that one, many of the isolates that we have fail on that, because the lactobacillus produce a lot of enzymes, and protease is special. And many of these enzymes can digest gelatin. But we don't want to digest gelatin, because if you digest gelatin, you are cleaving the mucins that you have in your gut. So you don't like that part. You don't like to digest gelatin, so we just look for the ones that don't digest gelatin, that have negative to gelatinases. Okay, and we continue, for example, for the production of polysaccharides. So I don't know if it's possible to see the blue blots. That blots means that bacteria have polysaccharides, and not all the bacteria. We test a batch of bacteria. We have 96 bacteria there, and just few of them produce polysaccharides, and just 3 of them have appreciable amount of a good amount of polysaccharides. So this is really important, the production of polysaccharides, for the attachment of bacteria to our gut epithelium. So this is something that we must look like. And finally, for the resistance to antibiotics. Some of these lactobacillus are resistant to antibiotics, and we don't want to improve in our gut the bacteria that are resistant to antibiotics, because if they can start to share information each other. Okay, so we just want ones that are not resistant to antibiotics. Okay, so this is last test, but not is the least, but not the last. The last one is the positive effect again, pathogenic bacteria. So we can test. I don't know if it's possible to see. Here you have a circle. You saw? You can see? Okay, that's circles. In that dots, we put the proteins that the bacteria produce. So we are cultivating the bacteria, and we obtain the supernatant, and we test the supernatant. And the idea is to see if the bacteria have capability to kill the pathogenic bacteria or not. Not the bacteria itself, the molecules that the bacteria produce. This is the idea. So in each of these dots, we have one isolate. We put the supernatant of the culture of the bacteria. So we test for different strains, including Bacillus, Salmonella, Vibrio, Clostridium, Escherichia, Streptococcus, so something that we don't want to have in high amount in our gut. And we show that the isolates, some of our isolates have that capability to kill, to destroy that pathogenic bacteria. So this is really, really important. So this number of tests is just to see, to show, to prove that we have something that have really effect. Because one thing is to say that the kefir is already known that is a probiotic, so the ones that we have is also probiotic. This is one thing. Other thing is to show. And the idea of Marcus every time is to show the things, and I appreciate a lot of this position in the market, and we have the things, we have the product, and we show the effect of the product. This is the idea, and here we show what we have in the kefir that have been produced. Okay, and we are not still happy, because the researchers are never happy. And we got some of that bacteria to sequence. We have a whole genome sequence of 2 of these bacteria, the ones that are more promising. And we found about 2,200 genes. So many of the genes are really important for the properties I'm already talking about, the antimicrobial properties, the attachment activity, and so on, the immunomodulation. And of course, we found a huge repertoire of carbohydrate-active enzymes. This is really important, and this is the reason why when we have the kefir, we don't have casein. We have peptides from casein, because that bacteria can degrade the casein and can degrade also the sugar that we have in the milk. What kind of sugar we have? Lactose. And sometimes the lactose is not good for us. And the beneficial part of the kefir is the lactose does not exist at all, as we know, because that bacteria produce a number of carbohydrate active enzymes that can degrade the lactose in the small sugars that can be absorbed very well. Okay, but we identify many of the genes that we already discussed: the polysaccharides, biosynthesis, the production of vitamins and folate, the production of GABA, the production of butyrate. Butyrate is also really important as immunomodulation agent, and of course the bacteriocins, different kind of bacteriocins. So in summary, we have a novel fermenting donkey milk that I don't know if it's possible to say that is a kefir, because kefir is a really specific microbiome that is already established in the market. Okay, this is different from kefir that already exists, so I don't know, but this is the job of the Marcus, it's not mine. So we have a new fermented product that has a huge potential as a probiotic, so health benefits. And that fermented have at least 87% of bacteria with probiotic potential. That is what we show, and we are really appreciate the work that we have done. And I thank you again, Marcus and Sandra. Thank you, all of you.